You're on.
All right, hello everybody. This is the group questions for lesson 5. Since questions 1 through 4 are pretty much the same, we're just going to go the overview of how to do these ones. And I have a template here on PowerPoint that'll help you walk through each of them. And alrighty. So first off, kind of step 0, I like to get the different alleles and what they mean just so I can kind of process it in my mind. The next step, we need to set up the trihybrid cross. And so we just have a heterozygous female mating with another individual, and it equals the table in question 2. For step 2, we need to find the allele arrangement in the heterozygous parent. And to do this, we find the 2 biggest phenotypes that have the largest progeny, and for each of those that it gives us in the table, we know that that's the arrangement in the heterozygous parent, and we just put a line in between them so that they can be, so it will give us the allele arrangement. For step 3, we need to determine the gene order, and so to do this, we need to find the 2 least abundant progeny, which are these ones down here.
Where'd he go?
I think he froze.
Kyle!
Man, this is not working out today.
Aww. Well,
do we go to question 5 then?
sure. Question 5.
Alright. So this is question 5 right here. This problem is talking about, or pretty much is just giving us a couple scenarios and asking us to kind of figure out what's going on with it. What are the different things going to do with the different scenarios? So the first one's talking about, suppose a genotype is, capital A, little a, or heterozygous for both alleles, and then it talks about what are the genotypes of the gametes produced by the cell, what's the frequency of each of these gametes. And so it's assuming that these are on separate homologous chromosomes, so pretty much it's the basic thing of what we were doing before we learned about this stuff. And so it should just be, what I'm assuming is a capital A, big B, or capital A, capital B, capital A, little capital B, could all show up in a 1-to-1-to-1 ratio. This is what the guy at if you watch the MIT video, that's kind of what he went through. And then he talked about having these traits linked to each other. And then this is kind of what was seen with everything outside of pea plants and fruit flies. And so they see that the capital letters were linked and the lowercase letters were linked. So they would only see these 2 genes. So you have a 1-to-1 to 0-to-0. And that was really perplexing for a lot of people until that one sophomore at Columbia figured it all out in an evening. And then it's talking about, and then goes to the next one. Suppose you have a cell that has a genotype heterozygous again, with A is linked to B and little a is linked to little b. What are the genotypes of the gametes produced by this cell if crossing over occurs between big A, little a, and big B, little b? And so what I assumed was this is what was going on with the pea plants, is that they were having the same one, so you would have the same thing as if it was the original one with A, and so you have 1-to-1-to-1 ratio. And now this is where it throws me because it's talking about pretty much the distance, saying that the distance between the alleles for big A, little a, and big B, little b, and that the alleles are 7.8 units apart, and pretty much that means that they're linked. And so the frequency of each of these gametes. So I'm assuming, I was thinking it'd be like this, but now that I know that they're linked, I almost think that it's supposed to be like this, and we would only see these alleles here. So we definitely need to get some more knowledge on this. Do you guys have any input about this stuff here, or does that make sense, or does that seem a little off to you like it does to me?
C and D is a little off. I think for D, you should have starting with C, because big A and big B are linked, and little a and little b are linked, and then it's saying that the crossing over happens on the A's and the B's, then you would have it, instead of like in B with big A and big B, you would have a little a with a big B, and then a big A with a little b.
So you'd have a big A with a little b,
and
a big B with a little a.
Yep.
Then those would be your phenotype. So you'd still end up with the 1 to 0 phenotype then. Is that correct?
I believe so.
Okay.
And then
so that's D.
And then on D, this one confused me for a little bit, but this is the difference between A and B, the distance. And in the reading, it talked about that most genes, if they're further than 50, which a lot of them are further than 50 mu, then the crossing over happens and then happens again, so it looks just like the parents. So it gets counted. And so on this one, it's talking about these are closer together. The linked genes are close. So if one switches, it most likely the other will switch as well. Because 7.8 is really, it's actually 0.078% to have that crossover happen. Which means if you do 1 minus 0.078, that gives you a 92.2% that it will stay linked with big A and big B and little a and little b. Does that make sense?
So you say that answer is correct then?
I say that answer would be correct if you put a 92.2% next to it.
Percent, you say?
92. 92.2. Yeah.
And then that's number 5.
All right. Do you want me to do mine again?
Yeah.
Because I didn't realize that I cut out when I was still presenting to nobody.
Oh no! Yeah. We cut out right on problem B when you were talking about using the DCOs to figure out what it would be.
Okay.
So slide number
was
4.
Was I right here?
You're on slide number 4.
On slide number 4?
Yeah.
Alrighty. Alright, so we're going to use the double crossovers to find out which gene is actually in the middle on the chromosome. And see, these are our double crossovers, and we need to use the heterozygous parent and find out which gene will go in the middle to produce are double crossovers. And for this one, we find out that if we switch the SC and the SC plus, then that will produce a double crossover. So we know that the SC is going to be in the middle. What the middle gene is now. Double crossover numbers, and then our single crossover numbers found with these ones. We'll use our mu equation to get 33 mu to. And then for step 6, it's the same, except you do the crossovers from the other side of the chromosome, which would be the V side. And we then will find the MUs. It's 10 MUs from the V to the center gene. For step 7, we need to make a map. From S to C, it's going to be 33, and then from C to V, it's going to be 10. And that's pretty much how you make a linkage map. It's pretty easy. Step 8, we need to calculate inference. First, we find the probability of the double crossovers. And we do this by taking our MUs divided by 100 and timesing them by each other. I'm not sure how that works. It just does. And we get this number right here, which we will use to find the expected number of double crossovers, which is 1,000 times the number we found, which is 33. So there's 33 expected and 24 observed. You plug that into the inference equation for observed over expected, and we get 27.3% inference. That's how you finish questions 1 through 4. And Ashley has something to say about question 3 because it's a little bit different.
So for question 3, if you can see this right here, I've got it set up. Right here, instead of the double crossovers, you actually do it with the smallest ones, which would be 0 and 0. And the reason for that is because there's only 6 phenotypes listed. So we need those other 2, and since they're not listed, they're automatically going to be 0. So the 0 and the 0 will be used for the double crossovers. And then you set everything up the exact same way through that PowerPoint. And now Noah will talk about problem 4.
Right, so on problem 4, you should fire the scientists that collected this data because their graph is all mixed up. They shift around what variables are in what order, and so you can write it down that way and go through the problem, but I found it easy to just take time to say this column is going to be for AN, this column is BR, and this column is F. And when you do that, it's a lot easier to read and a lot easier to help figure out. And that was the trick for number 4. Okay, and we talked about problem 5. So that would be all of them. Alright, I'm just gonna pause this recording.